Hannah Lutgen is the Floriculture and Landscape Junior Extension Agent for Maui County. Her programs include integrated pest and disease management, soil and plant health, and support of the floriculture and landscape industry statewide. She has a Bachelor of Science degree in Sustainable Horticulture and extensive work experiences as a conservation specialist, cut flower grower, florist, and landscape professional. Hannah's going to give us an ornamental update in Maui County. Hannah, thank you for being here.
Hello everyone, and thank you so much for having me. I'd first like to thank HFNA for coordinating this event, and would like to thank everyone online for attending, as well as everyone in this room. Okay, so today my main focus is to just give you an update on projects that I'm working on in Maui County. So I'll be talking about the Proteaceae pest and disease management, as well as what we're working on to maintain the germplasm, as well as the finished product, the import replacement project educational document. So the Maui Agricultural Research Center in Kula, Hawaii is home to a very diverse and large Proteaceae germplasm. And I first want to start by thanking my talented and wonderful collaborators, Dr. Lisa Keith, Dr. Roxana Myers, Lionel Sugiyama, Brian Bush, and their whole team at USDA-ARS PBARC, as well as the phenomenal Kula Agricultural Research Technicians, and a generous grant from Maui County Farm Bureau. We are currently trying our very best to preserve the germplasm collection for the current and future use to support the floriculture industry. So far, as Dr. Lisa Keith mentioned, there are over 20 fungal genera identified and isolated in Proteaceae at the Ag Station. We are currently working on perennial crop maintenance, so that includes pruning, weeding, fertilization, spraying with pesticides, and propagation. And we conducted soil and plant tissue testing to see what our soil is rich in and what we are lacking, which will really help us aid in soil and plant health, which I will talk about later. So these pictures here are a result of mulching with local freshly chipped Ficus benjamina wood chips. So we applied this about 3 to 4 inches thick to terrace 5, which, although it's a lot of work at first, not gonna lie, however, I think the benefits outweigh the cons in that applying freshly chipped mulches increases soil organic matter, which provides food for microbes and beneficial organisms in the soil, and it also helps to conserve soil moisture and suppress weeds. Currently, we are continuing our Proteaceae disease testing. As Dr. Lisa Keith mentioned earlier this morning, her talented team is working on in vitro fungicide assays at the PBARC lab, and we are working on a potted plant fungicide trials, which I'll go into shortly. This is all thanks to a grant from Maui County Office of Economic Development. So here in these pictures, you can see images from 2 commercial protea growers in Maui. So it's really important that we collect samples from across Maui so we can help understand, identify, and diagnose the pathogens that are affecting Proteaceae. This will help us better understand the diseases and help us come to decisions to reduce and manage these fungal pathogens. Okay, so we are currently conducting a potted plant fungicide trial, which is very exciting. In these pictures, you can see we selected a Proteaceae species and a Leucospermum pincushion, which were showing mild to moderate disease symptoms. Whoops. And we are testing 3 different fungicides to really evaluate, to mimic what we see in the field. So do these fungicides help increase plant health or reduce disease severity symptoms? This is the questions we are asking ourselves. So in this slide, you can see our experimental design. We have a randomized complete block design. And we are testing Pageant, pyraclostrobin, and Eagle myclobutanil in triathlon, which is actually a biofungicide. So it's a strain of Bacillus. We're applying them based on the label and collecting plant health, disease severity information, as well as monitoring weather, because as Lisa Keith mentioned earlier today, the disease triangle is very important, and weather and host and pathogen play a big role in disease. Okay, so based on the results, thanks to Lisa Keith and PBARC, the most important thing that we can do is prevent these fungal pathogens. This will help growers. This will help save the germplasm. And the ways to prevent these fungal pathogens is by keeping the plants in soil healthy. So there's other cultural practices we are testing, such as disease pruning, sanitation. Sanitation is critical. So every time you have your red Felco scissors and you're snipping, you're harvesting your flowers in the field, please be aware of what you're doing. If you see disease symptoms, if you go to one plant, if you're harvesting and then walk to another plant, you could be potentially spreading spores or disease to another plant, as well as creating an open wound where spores, fungal pathogens in the air, or that could be spread by splashing water, could infect that host. So please, please, please sanitize your tools and remove dead plants from the field. That's just a source of inoculum that could be spreading throughout your whole farm. It doesn't take that long. Just use a shovel and remove your plant. And if you're using fungicides, please make sure to rotate, follow the label thoroughly, and make sure to rotate both the FRAC code and the group. Okay, so future projects that we are working on are actually nutrient trials. So we are trying, based on the soil and plant tissue results, we are trying to use the correct amount of fertilizer, even using bioamendments and mulch to really stimulate the soil food web and provide the right amount of nutrients at the right time and the right application rate to help those plants stay strong and healthy. Another future objective is to actually interplant Nemagon marigold with the Protea to see if that helps suppress root knot nematodes in the Proteaceae terraces. So I'm really excited about these projects, and we'll keep you tuned next year. Lastly, I just wanted to touch on the import replacement plants, also known as IRP, the educational document that I created. The import replacement plants are a group of plants mostly in the Myrtaceae family. And if you're interested in learning more, you can refer to this document anytime. It contains information such as plant growth, plant habit, soil type, growth habit, and thanks to Chuck Chimera and BISC, it actually now contains the Hawaii Pacific Weed Risk Assessment Score, which is critical so that you are not planting something that could potentially become invasive. And the HPWRA, fancy word, that is basically a rating of a plant's potential of becoming invasive in Hawaii. So please pay attention to that. You can use this tool and click on it. The hyperlink is included. and it will go straight to plantpono.org, which is a great website full of all kinds of good information, and will have these scores which will help people decide what to plant and what's suitable in their area. So this is just an example of the Kunzea efficient, which is in Proteaceae family. And you can see the information. This is a disclaimer. All this information is based on the plant growing in its native habitat in Australia. So with that being said, it may or may not perform differently here in Hawaii, but please make sure to check out that HPWRA website and link. I would like to acknowledge all of my collaborators, so Dr. Lisa Keith, Roxana Myers, Lionel, Brian Bush, Kathy, and the whole USDA ARS PBARC team. I'm sorry if I missed some of your names, but I really appreciate all the work you've done. I also want to thank our wonderful agricultural technicians in Kula. I couldn't do it without your help. Really appreciate you. Havla Mills, my research technician, Dr. Tessie, and the entire Ornamental CTAHR extension team. If you have any questions, please make sure to email me at hannahc@hawaii.edu. And I just want to give a special thanks again to HFNA, Maui County Farm Bureau, Maui County Office of Economic Development, and PBARC. Anyone have questions?
Yes. Thank you, Hannah. And we do have some questions here. It looks like you have an amazing range of proteas. What can folks do to support your work with this collection?
I appreciate all help, especially because it is a large collection and we need continual funding. Like James Keach mentioned, every single agricultural research station is lacking technicians who do the majority of the work. They're the people in the fields, getting dirty, planting everything. They are amazing. We can't continue to do our research without them. So please make sure to contact me, and I would love to talk.
And it looks like, with your work on that fungicide, the fungicide research with the proteas, at one time there was a extensive industry in Hawaiian Ocean view estates in which, unfortunately, the volcanic fog, wiped out that industry in the southern portion of the Big Island. And so hopefully, your work will help to reestablish the proteas there as a viable industry. So good luck, and we look forward to the results of your research.
Thank you so much.
Thank you.
Thank you.